Gene expression can be controlled at many levels in eukaryotes, as illustrated in the graphic of this cell. Firstly, because of their more complex genomes and organisation of the genetic material, but also the fact that transcription and translation are separated by the nucleus, and mRNA must be processed before it can be translated. We're going to start at the level of chromatin. DNA is wrapped around histone proteins to form the protein-DNA complex called chromatin. The condensed nature of chromatin helps it regulate gene expression, as RNA polymerase cannot access DNA when it's condensed. To begin transcription, DNA near the promoter must be relaxed from its tight interactions with histones and other proteins. This process is called chromatin modification. Histones bind to negatively charged DNA because they're positively charged. The amino acid tail of histones can be acetylated by the addition of acetyl groups to relax the charge and open up the chromatin structure for transcription. The addition of methyl groups to cytosine bases in DNA is called DNA methylation. It's usually associated with reduced gene transcription. It can cause short or long-term inactivation of genes. For example, long stretches of inactivated X chromosome in mammalian females are heavily methylated. The pattern of modification of DNA in histones can vary from cell to cell and generally remain stable during mitosis. Nevertheless, these patterns can be influenced by the environment, for example, an individual's diet and whether they smoke cigarettes. Although the patterns are reprogrammed during gamete production and in the early stage embryo, some can be passed on to future generations, so the effects of poor diet and smoking during pregnancy, for example, could potentially impact 3 generations. This is termed epigenetic inheritance, where patterns of inheritance are not due to differences in gene sequences, but instead due to differences in DNA or chromatin modification. Diseases with a genetic basis seen in one identical twin and not the other may be due to epigenetic mechanisms such as this. Now let's reflect on the learning objectives we've covered so far. You should now be able to explain how DNA methylation and histone modification affects chromatin structure and the regulation of transcription at this level. And you should be able to describe what is meant by the term epigenetics. Once chromatin has been modified for gene expression, the next step where expression can be controlled is at initiation of transcription. We're now going to look at the types of DNA sequences called control elements and the types of molecules that bind to them for initiation of transcription. Every eukaryote gene has a promoter where RNA polymerase binds and starts transcription, proceeding downstream. Transcription in eukaryotes is regulated by a number of additional control elements, DNA sequences located near to or far from the promoter that act as binding sites for molecules called transcription factors. The distal control elements far from the promoter can be grouped together as enhancers, while the proximal control elements are located near to the promoter. Control elements and the transcription factors that bind to them are critical for precise gene regulation in different cell types. The cell-specific expression of genes is dependent on 2 factors: the difference in chromatin modification and the presence of different regulatory proteins called transcription factors, which bind to control elements in the promoter. There are 2 types of transcription factors: general or basal transcription factors that interact with the promoter, and specific or regulatory transcription factors that bind to enhancers and other control elements to control the expression of specific genes in particular cell types and at particular stages of development. Specific transcription factors can be activators or repressors. Different cells have different transcription factors which are produced in response to signals that occur during early embryonic development. These transcription factors enable the different cells to have their unique characteristics. For enhancer regions far from the promoter to influence transcription, they must influence the promoter. Mediator proteins make up the mediator complex that assembles and positions the initiation complex at the promoter. By doing so, the mediator complex creates a physical link between specific transcription factors, general transcription factors, and the promoter. Here we see cell-specific transcription in liver compared to lens cells. Both the liver and lens cells each have genes for making the proteins albumin and crystallin. Albumin is one of the major proteins in the blood and is produced by liver cells. Crystallin is a protein that makes up the lens of the eye and is produced in lens cells. These cells produce different proteins because of specific transcription factors in each cell that are different, and these transcription factors determine which genes will be expressed. We can see that each gene has enhancer regions of 3 different control elements that are made up of a unique combination of elements. Certain activators bind to the enhancers for the albumin genes and promote the expression of albumin in liver cells, while a different set of activators in the lens cells bind to the enhancers for the crystallin genes and promote the expression of crystallin. As a result, each cell produces cell-specific proteins. Chemical signals from outside the cells also regulate gene expression in eukaryote cells. For example, steroid hormones are lipid-soluble and can diffuse into cells, bind to intracellular steroid hormone receptors and form a hormone receptor complex. These complexes can then bind to DNA at specific sequences called hormone response elements, and they act as transcription factors. The hormone receptor complex can interact with general or specific transcription factors, turning genes on or off. The same hormone can affect different cells. For example, estrogen affects expression in many cells of different genes, from reproductive to cardiac cells. We saw how prokaryotes use operons to express genes involved in a similar function or pathway. Eukaryote genes each have their own promoter, so the operon strategy wouldn't work. But recently it's been discovered that they can physically associate with other genes by looping their chromatin into regions of the nucleus rich with RNA polymerases and transcription proteins. These regions have been termed transcription factories. Now let's reflect on the learning objectives we've covered so far. You should be able to distinguish between specific and general transcription factors. Should be able to explain the role that regulatory sequences and control elements play in transcription control. You should be able to describe how steroid hormones can influence gene expression and describe the role of proteins that make up the mediator complex. You should be able to explain how transcription factories help control related genes and, importantly, note the difference between transcription factories and transcription factors. Once DNA has been transcribed into mRNA, a series of events must occur if the final product is going to affect the cell. These events are mRNA splicing, blocking or altering translation rates, and modifying the lifespan of mRNAs and proteins post-translation. During splicing, introns are spliced out of the primary mRNA transcript before it leaves the nucleus. In alternate splicing, changes in gene expression are possible because selected exons can be removed from the primary transcript along with the introns. As a result, the same primary mRNA transcript can yield more than one kind of mature processed mRNA consisting of different combinations of transcribed exons that can lead to the production of different proteins. The tropomyosin gene codes for proteins that are associated with smooth and skeletal muscle cells. So here we see that the primary mRNA transcript can be spliced in 2 different ways, generating different mRNA molecules, one with exon 3 and not 4, and the other with exon 4 and not 3. These 2 mRNA molecules are translated into different but related muscle proteins. Humans have approximately 25,000 genes, and we can produce at least 40,000 proteins this way. Initiation of translation provides another opportunity for regulating gene expression. Regulatory molecules can bind to specific sequences or structures within the untranslated region of the mRNA, preventing the attachment of ribosomes. Alternatively, translation of all the mRNA molecules in a cell can be regulated simultaneously. Such control usually involves activation and inactivation of molecules that are required for initiating translation. This mechanism starts translation of mRNAs that are stored in eggs. Just after fertilisation of the egg, translation is suddenly triggered by a sudden activation of translation initiation factors. The lifespan of mRNA molecules in the cytoplasm is important in determining protein synthesis in a cell. Eukaryote mRNA is more long-lived than prokaryote, and survival usually depends on the cell's requirements. Nucleotide sequences that influence the lifespan of mRNAs in eukaryotes reside in the untranslated region at the 3' end of the molecule. An example is the control of mRNA degradation in response to iron in red blood cells. If cells have low iron stores, an active iron response element protects the mRNA molecule from degradation. The mRNA molecule, protected by the active iron response element protein, codes for a protein called transferrin, which is responsible for transporting iron into the red cells. Once iron levels are high, iron binds to the iron response element, deactivating it. So as a result, the mRNA that codes for transferrin is degraded. Just 1.5% of the human genome is protein-coding DNA. The remainder used to be called junk DNA, but we now know that much of what we thought was junk can be transcribed into non-coding RNAs. Non-coding RNAs appear to regulate gene expression at 2 points: mRNA translation and chromatin configuration. There are several classes of non-coding RNAs. At least 20% of all animal and plant genes are regulated by these non-coding RNAs. MicroRNAs are single-stranded RNA molecules that can bind to complementary sequences in the mRNA to either degrade the mRNA or block its translation. Another class of non-coding RNA is small interfering RNAs. As studied in yeasts, they appear to play a role in heterochromatin formation and can block large regions of the chromosome. Finally, after translation, many proteins undergo chemical modifications such as cleavage of amino acids or addition of carbohydrates to become functional. Regulatory proteins are commonly activated or deactivated by reversible addition of phosphate groups. Chaperone proteins can regulate folding of proteins to regulate their functions. Another method of control is targeted protein destruction. Ubiquitin tags are added to proteins like cyclin, which are recognised by a proteasome, which cuts the proteins into short segments. Now let's reflect on the learning objectives we've covered so far. You should now be able to describe the importance of control of gene expression in both prokaryotes and eukaryotes. You should be able to describe at what levels gene expression is generally controlled in eukaryotes, and as we see, at many levels. You should be able to describe why the control mechanisms are different in prokaryotes and eukaryotes, and think about the cell and the fact that eukaryotes have a nucleus. You should be able to explain the process and significance of alternate RNA splicing. You should be able to describe factors that influence the lifespan of mRNA and those that bind to and degrade to mRNAs. You should be able to explain RNA interference and the role of microRNAs and short interfering RNAs in gene expression control, and describe the role of protein processing and degradation in control of gene expression in eukaryotes. Thank you for watching.